In this video, we are talking about the reproduction in Sycon. So students, first of all, I will be giving you the basic two types of reproduction in Sycon. So the very first type, it is asexual type of reproduction. So this is the very first type. So this asexual kind of reproduction can happen in Sycon by budding. So this is the process by which the asexual type of reproduction it happens in case of Sycon. So what happens that in budding an outgrowth is formed along with the cylinders on one side, and that particular bud that grows in size, it forms its outpore, the osculum, and this particular bud now it has got two fates. So first fate can be that it can remain attached to the parent Sycon. So the very first fate it can be that it can remain attached, and the second fate it can be that it can detach from the parent and can swim to some extent and can go away from the parent, and that cut off bud it can make a new Sycon as such. So these are the two fates of the bud in the Sycon. So this is a kind of asexual type of reproduction. So it involves only the single individual. A bud will be formed, that will be an outgrowth, and that outgrowth can remain attached to the parent Sycon, or it can detach itself to make a new Sycon. So this is the very first type of reproduction, that is the asexual reproduction. Now after asexual reproduction, we have got the second type, which is the sexual reproduction. So Sycon, it is also showing the sexual kind of reproduction. Now first of all, in this part, I should explain it to you that the Sycon, they are monoecious kind of individuals. So monoecious refers to that both the male and the female, the organs they are present in the same individual. So the Sycon, as it is producing the sperms and the ova in their mesoglia, the centralmost layer, so we can say that they are monoecious in nature. So each Sycon has got both the ova and the sperms in the mesoglia. But the fertilization is of cross type. So as the Sycon has got both the ova and the sperms, but the sperms of a Sycon cannot fertilize their own ova, the ova of their own Sycon. So there are special mechanisms that the sperms they travel to the second sponge, and then they travel up to the ova of the second sponge, then they can fertilize, as we have seen the cross kind of fertilization in the Sycon. So first of all, uh, we should talk about the organs or the gonads. So there are no special gonads in case of Sycon, as we have seen in the case of in the previous videos, that there are no special organs. So the sperms and ova they are formed inside the mesenchyme. These are the gametes. So gametes are formed in the mesenchyme, the centermost layer of the Sycon, and these are formed from archaeocytes. So that means archaeocytes they differentiate to form the gametes. So now, where are these gametes present? They are present in the mesoglia. So this is the middle layer, this is the innermost layer. Now, if we consider further, now the sperms from one Sycon, they are going to the another Sycon. So if I go with the first process, first of all, this is spermatogenesis. So spermatogenesis is the formation of the sperms, and this process, it starts with the sperm mother cells. So here we write them as SMCs. So a sperm mother cell is a large archaeocyte which is present in the mesoglia. Now it is surrounded by one or more cover cells. So these are the cells which are to nourish this particular sperm mother cell, and cover cells are also formed from archaeocytes. This is a very good feature of the archaeocytes, that they can make any kind of cell in the body of the Sycon, as they are undifferentiated cells. So the SMC, the sperm mother cell, and the cover cells, they are all formed from archaeocytes. Now when the SMC, it is covered with the cover cells, both they make spermatocyst. So the structure which is formed with the SMC along with its cover cells, it is called as spermatocyst. Now this spermatocyst, it divides 2 or 3 times. So what it is dividing. So when it divides, it forms many spermatocytes. So after division, what it is forming spermatocytes. So these spermatocytes, they are ultimately forming spermatozoa. So all this process from SMC up to the formation of spermatozoa, this is altogether occurring in the mesoglia, the centermost layer. So as we have discussed earlier, that in the mesoglia we had the gametes. So where are the gametes? These are the gametes. So these are present in mesoglia, and the process is called as spermatogenesis. So this is the formation of spermatozoa. And now if I go back and come to the second phase, the second phase refers to the oogenesis. So this is the formation of eggs. Now the formation of eggs, it starts with the egg mother cell. So we write here it as EMC. So this is the egg mother cell, and many authors, they write it as oocyte. So egg mother cell can be written as oocyte. Now oocyte, or the egg mother cell, is also formed from archaeocytes as the sperm mother cell. So the case is the very same. So what the EMC, or the egg mother cell, it is doing, it grows in size. First of all, it grows in size, and it shows 2 maturation divisions. So it is undergoing 2 maturation divisions. So after these maturation divisions, we have the final ovum. So this is the process of the formation of the ovum. So this ovum, it lies again in the mesoglia, and it is lying near to the ovum this is a very important feature of the ovum. The ovum is lying near to the choanocytes, as we have earlier discussed in the previous videos of the function of the choanocytes, that the choanocyte is helping in the fertilization process. So that's why the ova, they lie near to the choanocytes in the radial canals. So this is again a very important point. Now these ova, they are ready for fertilization. So what they need is the sperms of the other sponge, because this particular sponge has the sperms also, but those sperms are unable to fertilize these ova. So they need the sperms from the other sponge, as we have the cross fertilization. So we are continuing with the sexual reproduction. So after spermatogenesis, that is the first step. Second step was oogenesis, the formation of eggs. Now the third step is fertilization. And this process is internal and cross. So this is a very important feature, that it is internal and cross. So as we have earlier discussed, that this is a cross fertilization. So now suppose this is Sycon number 1 and this is the Sycon number 2, and this is the Mesoglia. So if we take this particular membrane as the Mesoglia, now this Mesoglia has got both the sperms and the ova. So if I take, suppose this is the sperm of the Sycon number 1. So we are taking, this is Sycon number 1. This is Sycon number 1, this is the Sycon number 2, and these are the sperms of Sycon number 1, and this is the egg of Sycon number 2, and this is the sperm of second sponge, and this is the egg of sponge number 1. So as we have discussed earlier, that every sponge has got both the sperms and the eggs. Now what will happen, that the sperm from Sycon 1, it will come out into the water, and this sperm will enter into the ostia of Sycon number 2. So it will go into the Sycon number 2. So as we have discussed, that the food goes from ostia, then to incurrent canal, then to radial canals, and then to the spongiocele. So finally, as we have previously discussed in the video for the choanocytes, we have discussed the whole together the physiology of the fertilization. So actually the sperm from the first will go to the sponge number 2, and then it will fertilize the egg of number 2 sponge. So that will be a cross kind of fertilization, and this particular sperm, the yellow sperm of Sycon number 2, will fertilize the egg of sponge number 1, or it can go to sponge number 3 or 4. So this is the cross fertilization. Now if I brief about the whole of the process of fertilization, it was that the sperm, it entered into the choanocyte, which is very near to the ovum of the second sponge. So as we have previously discussed this in the previous lectures, so it is entering into the choanocyte, the sperm is losing its tail, it is becoming a round kind of a structure, and the head of the sperm, it is entering into the choanocyte. The choanocyte, it is also losing its color, it is losing its flagellum, so it is becoming a round cell, and that cell is called as the carrier cell. Now this carrier cell is received by the ovum by the formation of a reception cone. Now that particular sperm will enter into the ovum, and then the ovum will be fertilized, and a zygote will be formed in the mesoglia. So that was the process of fertilization. Now after fertilization, what we have is the formation of a zygote in the mesoglia. Now next we will be starting up the development of that particular zygote. So if we go further, now after fertilization, what we have the zygote in the mesoglia. Now the next thing that we have is under the sexual process, we start from a zygote and we start with the development. Now under development we have got 3 periods. So we have got 3 periods. So this is the first period, this is the second period, and this is the third period. The first period is also called as the early embryonic period. So this is the early embryonic period, very start from a zygote. The second period is known as the larval period, and the third period is called as the post larval period. So these are the 3 periods in the development of the Sycon. So first of all, we should start with the first period, that is the early embryonic period. So if we start with the EEP, first of all we have got the zygote. Now this zygote, it undergoes the very first part of EEP. It is again divided into 2 subparts. So first part is the cleavage, and EEP's second part is called as the stomoblastula. So this is the second stage. So if we start with the very first part of the EEP, which is the cleavage, so we start with the zygote, and this zygote, it undergoes 3 vertical divisions. Now when it will be cut 3 times vertical divisions, we have 8 blastomeres. So the first division will give you 2, second division will give you 4, and third division will give you 8 blastomeres. So after 3 vertical divisions, we have got 8 blastomeres. Now these 8 blastomeres, they undergo the second division, but this division is horizontal. So this is a very important part. So we write here as 1 horizontal division. So this horizontal division will give you how many cells? So they will give you 16 blastomeres, but they are in 2 tiers. That means they are in 2 layers. So just imagine, okay, you have 8 blastomeres and you cut them horizontally. So it will give you 8 blastomeres on the top 8 on the lower side. So you've got 16 blastomeres in 2 tiers. Tiers means layers. Now out of these 16, we got again bifurcation. The upper 8, they are smaller in size. That means 8 are micro and lower 8 are macro. So out of these 16s, we have got 8 micromeres and 8 macromeres. And 8 micromeres, they are the upper ones. And 8 macromeres are on the lower side. So that's why I have written it towards a lower level, because 8 are present on the lower side and 8 are on the upper side. Now this is the condition up till now. Now we are emphasizing on the upper 8 micromeres. So these are the upper 8 micromeres, which are smaller in size. That's why they are micromeres. So these 8, they divide mitotically. So they show mitosis, they divide. But 8 macromeres, they are not dividing. So you should emphasize on this point. So the upper 8 micromeres, they divide mitotically and they form many cells. So they are dividing and they are forming many micromeres. So due to the division of the 8 micromeres, we have got a number of micromeres, and this makes a particular structure. After division, we have got a structure which is called as blastula. So this is a round structure, this is a ball-like structure, and it is having a cavity inside it, the blastula, and the cavity, it opens on the lower side. So just like a pot which is like having a mouth, but the mouth is on the lower side. So this is the structure of the blastula. So this blastula is due to the formation of these 8 micromeres, the division of the 8 micromeres. So this is a kind of blastula which is having a mouth on the lower side. This is the mouth, and the mouth is surrounded by cells which are the macromeres. So here we have the 8 macromeres. 8 macromeres on the mouth. They are present on the periphery of the mouth. And here we have this part is all composed of the micromeres, because micromeres, they have divided into many cells. So the blastula, the 95% of the body of blastula, it is made up of micromeres, but we have only 8 macromeres on this particular rim of the mouth. So this is the first stage, which is called as the blastula stage, and some people, some scientists, they call it as stomoblastula. So you may get this question that what is stomoblastula? So it is also called as stomoblastula. Stomo refers to mouth. So this is the blastula which is having a mouth on the lower side, which is surrounded by 8 macromeres. So this is the structure of the stomoblastula, and this is our second phase. As the early embryonic period, it was divided into 2 parts, the cleavage and the formation of stomoblastula. So at the very end, now we have the stomoblastula formed inside the mesoglia. So altogether the whole process is going on in the mesoglia of the Sycon. Now, if we take the stomoblastula into account, one side has got many micromeres. On the upper side we have got many micromeres, and on the lower side we have got how many macro? These are 8 in number. So we have got 8 macromeres on the lower side. So it opens by a mouth, and after some time you should be very clear that the micromeres which are present on the top, they all bear flagella. So they form flagella, but the flagella, they are on the inner side, so they are not visible on the outside of the blastula or the stomoblastula. So if I make suppose a TS of this stomoblastula, it will be just like this. So this is TS or VS, you can say. So this is one macromere, this is another macromere. So we have got 8 macromeres in the circle, and here we have many micromeres. So these are all the micromeres, and these micromeres, they are having flagellae towards the inner side. So they are bearing flagellae, but they are towards the inner side of the stomoblastula. They are not seen towards the outside. So this is the case. So that is the second stage, which is called as the stomoblastula. So students, I think you are very much clear about the very first phase, the early embryonic period. So this is all the process of early embryonic period. Now we start with the second period, which is the larval period. Now in this period we start with the phase which is called as amphiblastula. So this larval period is again divided into two subphases. First phase is the formation of amphiblastula. So amphi refers to both, so we'll be taking out later on. So the second phase here is the formation of gastrula. So these are the two phases which are present in the larval period. So first of all, we are taking up the very first phase, which is the formation of amphiblastula. So till now what we have is the stomoblastula. So the structure which we have discussed in the later slide, that was the stomoblastula. So we start with the stomoblastula. Now it undergoes a particular process which is called as inversion. So what happens, that the whole of the stomoblastula, it inverts out. So what is present inside is now inverted out. Now all the flagellae which were present towards the inside, now they are towards the outside. So now if I again draw one diagram of this particular stomoblastula, or here I should say the amphiblastula. These are the 8 macromeres on the lower side. So on the top we all have the micromeres, but now these micromeres, they have the flagella towards the outside. So that means you have got a huge number of flagella which are protruding towards the outside of this particular larval stage. Now this stage, the inverted stage, it is called as the amphiblastula. Now why it is called as amphiblastula? Because it has got both the type of the cells. Half of the cells, or you can say it is also having the flagellated cells and non-flagellated cells. So that's why we refer to as amphi. Amphi means both. Both refers to non-flagellated are the megameres, the 8 megameres, and the flagellated they are the micromeres, and they are huge in number. So that's why it is called as amphiblastula. Now this stage, it is present in the mesoglia of the sponge. Up till now, the development was in the mesoglia of the sponge. But now this amphiblastula, because it has got now flagellae which are towards the outside, this stage it comes out of the sponge. Now this stage is free in water. So this is a very important point, and it is coming out of the sponge, and it can perform the swimming movement. So it can swim because it has got a number of flagellae. Now we start with the second phase, which is the formation of the gastrula. So what happens that this amphiblastula, now it is free in the water, it swims to some extent, it can go to some distance, and after swimming to some distances, it performs gastrulation process. So what happens when it swims, then it settles down. So it settles onto the substratum. When the macromeres they touch the substratum, so the touch it stimulates the division again, and it starts the formation of the gastrula. So when the amphiblastula it swims to some distance, it settles down. When it touches the ground or the substratum, then a division occurs, and now the macromeres will divide. So this is the important thing. So after settling down, now these macromeres, which are 8 in number, now they will divide. So this is a very important feature. So after settling, it performs the process which is called as gastrulation. Now what happens in this gastrulation, that the macromeres they divide rapidly, very furiously, and they cover the whole micromeres on the top. So when they divide, just see the diagram, when they start dividing, they will cover, I should take it a red color, so they will cover the whole of the micromeres on the top, just like this. So now this gastrula, which is formed after the division of the macromeres, now it is two walled. That means it has got two walls. Inside wall is of micromeres, outside wall is of macromeres. Now where are the flagellae? Now flagellae, they are again coming towards the inside. So this is again a very important point. The flagellae, they are again coming towards the inside. So these are the flagellae. So now this is the structure which we call as the gastrula. So this is the phase of gastrula formation. So students, at the last, now we have the gastrula, which is a two-layered structure. Inside we have got the micromeres, a number of micromeres. Outside we have got the macromeres. Now the inside cells, they have got the flagellae. Now we start with the third phase of the development, which is the post larval period. So this is the very last period of the development, and this period is also called as metamorphosis. So this is a very the process which is involving a huge change in the animal. So that's why it is also called as metamorphosis. Now this gastrula, here we have written as amphiblastula, but this is not the amphiblastula. Now it is this is the gastrula. So this particular formation, the foam, it is settling down. Now gastrula, it settles down. First of all, it lengthens and it becomes like a cylinder. So it becomes elongate and it forms a cylinder like structure. So we should start with gastrula. It elongates, it forms a cylinder like structure, and it forms an osculum on the top. So it forms the osculum. So osculum is formed on the top, and the non-flagellated, which are present towards the outside, these cells, the non-flagellated, these are the macromeres. They are forming the pinacoderm. So this is the outermost layer of the Sycon. If you remember, the Sycon has got 3 layers. Outer was the pinacoderm, inside layer, the innermost layer was the choanoderm, and the central layer was Mesoglia. So here the outermost layer is forming the pinacoderm. So we can also say that macromeres, they are making the pinacoderm, and towards the inner side the second layer was of micromeres. So because these are flagellated, they are forming the second layer, which is the choanoderm. This is the innermost layer, and the mesoglia will be formed later on in between these two layers. So at last we have developed the 3-layer system which is present in the Sycon. So at last 3 layers are formed, and this long cylinder like structure which is formed from the gastrula, it is called as the olynthus stage. And this is a very important question. So many of the times you get a question on the olynthus stage. Actually the olynthus stage is very similar to the Sycon, but there are no radial canals, there is no canal system formation, only the osculum is present. So this is the difference between the olynthus stage and the final adult Sycon. So after this olynthus stage, these are the characters of the olynthus stage. It has got 3 layers, it is cylinder like, it has osculum, but it has no canal system, or I should say the radial canals, to be very precise. So after this olynthus stage, after the radial canals are formed, I should write here, radial canals, they are formed, and it undergoes further differentiation. So after further differentiation, what we have is the adult Sycon, which is fully formed. So students, this is the whole life cycle of the Sycon. So I expect that you have understood this particular topic. See, this is long enough, but if you go by this cycle very systematically, so there is no problem in this life cycle. if I recall whole of the life cycle, it starts with the spermatogenesis, oogenesis, then we had the fertilization, it makes a zygote inside the mesoglia, then the development starts. The very first period is the early embryonic period. It is involved in the cleavage. Zygote, it cleaved into 8, then to 16. Two tiers, above 8 are micro, lower ones are the macros. Above 8 micros, they divide mitotically and they form a blastula. It has got a mouth on the lower side, which is surrounded by 8 macromeres, and that is called as a stomoblastula. So this is the second phase. The second phase starts with the larval period. We have the amphiblastula stage with the inversion of the stomoblastula. The flagella, they come towards the outside. It comes out of the Sycon, it swims, and after swimming to some distances, it undergoes gastrulation by the division of the macromeres over it, and it forms the gastrula, which you can see in the right diagram. Now this gastrula, it undergoes the post-larval period. It lengthens, it becomes a cylinder-like structure, it forms osculum, it forms 3 layers, and this stage is called as the Eulenthus stage. It further differentiates, radial canals are formed, and it forms a full Sycon. So I hope you have understood this particular topic. Thank you.